Okay, good afternoon, everyone. After having a gap of one day every day, we are back to the class, and then science and technology. That is what. So I thought, like after 15th August, Independence Day, I should also have new clothes and new shirt and all this part. New page there. So I thought like blue in color, something, right? So let's come to the blue biotechnology. What is blue biotechnology? Marine areas, right? All right, now, last class we covered about genetic engineering, tools and technique in biotechnology. And I said that genetic engineering is also said to be gene cloning because we make multiple copies of a gene. And that happens through the, after making recombinant plasmid or phasmid, virus and DNA, there we do that. Now next what we'll cover, and then I'll come to one current aspect also there, cloning part of it. Technique of cloning. Now, in a general term, clone refers to identical individual. They refer to clone. But in biology and biotechnology, cloning comes in different aspects. So let us understand in this way. Okay, when we say cloning, I can bring into three types there. We can divide into three aspects there. The first cloning is called gene cloning, where we make multiple copies of the same DNA or gene, DNA sequence there. This refers to gene cloning. And mostly, why it is done? To make something called library of DNA. Right? Sometimes this is used for making DNA library. And basically it finds application in what I have written there, genetic engineering, recombinant DNA technology. Again, what I said from the UPSC prelims exam point of view, such technical words should be remembered. So that when multiple choice question, objective question there comes, you are able to recall what is in what context it is saying there. Now second type of cloning is called reproductive cloning. The word here is reproduction. You all are aware that even normal case of fertilization, when egg gets fertilized and zygote is formed, and the zygote breaks into two, then it gives rise to identical twins. But what is called homozygotic twins. Now they become identical. Why? Because the zygote is the same, chromosomes are same. So they share the same gene or DNA. So they become identical. But when you talk from this cloning perspective here, what is the reproductive cloning here refers? That is a identical individual to one parent, and it is not a sexual reproduction. Normally twins, identical twins comes through zygote, and zygote is produced through the sexual reproduction, fertilization of egg. But let's say you want to make an animal clone, right? Clone of an animal, that is similar to any of the parent, male or female, and that is done in the lab. We manipulate in the lab. So here it is based on, let's say, asexual reproduction, and it uses a technique, what is called as, which was asked this year exam, the technique is called a somatic cell nuclear transfer technique. I'll go to all the detail also, but first let me write the name of the technique what is used worldwide to make identical clones, like identical copies of and clones of animals. You all know that human cloning is banned on ethical grounds, but we can use the same technique and we can make clone of a human being also there, but it is banned on the ethical grounds. But other animals, worldwide cloning has been tried out. Now what comes the third one is something called therapeutic cloning. Third type of cloning is called therapeutic cloning. So I have used the word therapy, treatment. In terms of treatment part, therapy comes into that. Now what is the therapeutic cloning? It will be really inhuman and unethical that we make a clone of ourselves and then take kidney or lungs or heart from that person. Any clone is also a living person, right? That's what one of the reason comes that human cloning is banned for any medical grounds and medical use there, right? Or even let's say animal. We make animal and then take the organ or something of that. But there is something, we can use a method of cloning and without producing the individual, we can go for treatment of diseases, right? And it happens through something what is called stem cell, right? Stem cell. So I'll be coming to that part. Now what is therapeutic cloning? where here use of, one by one we'll be coming to that, but first let me write use of embryonic stem cells obtained from right, so use of embryonic stem cells obtained from what I'm writing a very important name, blastocyst. I'm using again a medical term, blastocyst. Now what is blastocyst? Once an egg is fertilized, like after fertilization, this when fertilization normal, which happens in human beings or animal and all fertilization part. So what happens in after fertilization is that the zygote is produced, and this zygote divides. It divides very fast and forms a mass of cell. What is called as this looks like a mass of cell. This particular this structure is referred to blastocyst. Now inside this blastocyst there is a cavity, right, which contains embryonic stem cells. Now what are stem cells? The stem cells are those cells which has got capacity to divide, to give rise to any kind of cells, or it can produce itself. Same type or any other type of body cells can be produced. Now this happens in the zygote stage, blastocyst. Now let's say you want you are suffering with kidney damage and you want to repair that kidney and you require kidney cells. Right? One way is out that through the method of cloning we can make our blastocyst. And from blastocyst we can take the stem cells, and from stem cells we can generate in the lab kidney cells. So this is called therapy. Using method of cloning to produce the blastocyst, because the blastocyst will have the cells what we have got. Right? That's what, because remember that stem cells are also unique to a person on the genetic ground. So let's say we want stem cells, but we don't have right now is embryonic stem cell. Where was the embryonic stem cell? When we were in the fetus of mother. At that time this stem cell was there. Presently in the body we have adult stem cells, which are very, very specific. Right? But we want this kind of stem cells, which are has more potential. Right? Pluripotential is there. So what we can do? We can use the method of cloning to make this kind of blastocyst. And from there we extract the stem cells. We'll be coming back to this concept, but let's first understand what exactly the technique of cloning is all about. Then I'll come back to the stem cell and how, which will help in therapy part. Now understand here. Each one of us may be similar to any of our parents. We'll have the similarities there, but will never be totally 100% similar to any of the parents. Because what happens? 50% chromosome from mother, half the chromosome from mother, half chromosome from father. They mix together in fertilization and forms the entire zygote. There is always a recombination. It happens there. But what we want a cloned animal, which is a cloned individual, which has all the chromosomes. So what is done here is that, now I'm coming to what was asked in the prelims exam, the name of the technique. I'm writing here. See, what happens is somatic cell nuclear transfer technique. So first I'll draw with a sequence of the steps, and then I'll give you a paragraph on writing part. What exactly scientists do in the lab, right? Again, remember that all this for understanding purpose. There in the UPSC exam, you will not be asked to write what is somatic cell nuclear transfer technique, right? But yes, in the state PCS exam, any other competitive exam, there when you write the mains exam, the direct questions are asked. Explain cloning, write cloning, and there you have to write. But in the PT exam or something will not be there. This is only for understanding. So what is done there? Let's say, for example, we want to make, let's say, an animal X. We want to make exactly same X animal there. So what scientists do? They take from the body something called body cell, somatic cell. See, in the human body, there are two types of cells. Even the animals also have what is called somatic cell. Normal body cells are called somatic. And there is one type of cell called reproductive cells. Reproductive cells are, germ cells are half chromosome hota hai, half chromosome. But somatic cell, body cells have all the chromosomes, right? So let's say we want to make a clone of this animal X. We want to make another X there. We take the body cell. Let's say, for example, if I'm writing here, skin cell. Just take example, skin cell. Now skin cell, this cell here, in the nucleus, where do you find chromosome? In the nucleus. This is where you have the all the chromosome of the organism. Now what is done? We take a female animal. But again, remember that when you're talking female animal, it has to belong to the same species. If it's cow, then cow. If it's dog, then dog. Camel, then camel. You take the camel you want to make and you make a dog, female dog, that's not possible. Same species, right? Now what is done here, we'll take from ovary. From ovary, there is something called germ cell, reproductive cell, egg cell, or ovum, that is taken, right? Let's say if I draw with the another diagram here, this egg cell is there. So egg cell will also have its nucleus. This nucleus is there.
But ovum would have haploid number of
half the chromosome, that is part of there. But now you understand there what will happen. We take the egg cell, it also has the nucleus. What is done? This nucleus is removed. In the lab, this is removed. Right? Now that means there is no chromosome from this animal. So now this cell will be totally devoid of nucleus. Right? And this is called what something called enucleated or denucleated. So let me use the word here, denucleated egg cell. It doesn't have its nucleus. Now what we can do? We want here all the chromosome. The person has to identify all the chromosome. So we can here either transfer or something, or we can do the cell fusion. We can fuse together both the cells. So let me write here transfer here. So this is called nuclear transfer technique. Right, so what will happen here? Now this egg cell which I am drawing here, this egg cell will be having the nucleus of this one. Right, this is what we have transferred. This part has been taken. Right? Now see what happens. Egg contain half the number of chromosome, what he has used the word haploid. Sperm contains half the chromosome, that is also haploid. In the fertilization, what happened? N plus N together form 2N, diploid. That's zygote. Now if you induce, what I'm writing here, it can be induced chemically in the lab, induced to divide like zygote. It can be induced to divide like zygote. Like zygote. It is not, remember, it is not natural. We are doing it in the lab. We are doing the manipulation in the lab. So we have to induce like a zygote. Now when this will divide, so what will form here? It will form what I've said, the mass of cell. Always this embryonic stage which happens here, blastocyst. The blastocyst is there. And this is can be transplanted. What I'm writing here? Transplanted into the uterus of the animal. Right? So what I've drawn here, I'll just explain here what exactly I meant to say there. Let's say we have to want to make a clone of the animal. So what has been done is that I have the scientist will take any body cell of that with animal to be cloned, right? Donor cell, right? Take another female animal. From that female animal, take the egg cell, ovum. Remove the nucleus of that ovum so that the egg cell is without its nucleus. Will have no chromosome of that animal. तो वो जो जहाँ से एग लिया गया है उसका न्यूक्लियस हटा दिया जाएगा तो उस न्यूक्लियस का क्रोमोसोम ही नहीं होगा, उस फीमेल एनिमल का कोई भी कंट्रीब्यूशन नहीं है, केवल क्या है उसका जो एग का जो पार्ट है वो बस केवल है. एंड देन व्हाट वी डू, वी टेक द जो स्किन सेल का जो न्यूक्लियस है, टेक दैट न्यूक्लियस विच कंटेन ऑल द क्रोमोसोम्स एंड ट्रांसप्लांट इनटू दैट एग सेल. तो अब जो एग सेल बन रहा है उसमें सारा टोटल क्रोमोसोम आ गया एंड देन कुछ केमिकल ऐड करके उसको इंड्यूस करते हैं डिवाइड करने के लिए, ताकि वो आर्टिफिशियली लैब में डिवाइड करके जाइगोट बन जाए और जाइगोट से ब्लास्टोसिस्ट बन जाए. अब ये ब्लास्टोसिस्ट को हम क्या करेंगे, यूट्रस में ट्रांसप्लांट कर देंगे, ट्रांसफर कर देंगे. हाँ, दैट्स व्हाट सेड सर्वाइवल, बिकॉज व्हाट इज वेरी इंपॉर्टेंट, यू हैव टू सी द सेम स्टेज ऑफ सेल डिवीजन. वहाँ पर बहुत सारे मैनिपुलेशन, ये तो मैंने एक जनरल एस्पेक्ट बताया. व्हाट आई सेड हियर इज दिस जनरल एस्पेक्ट हाउ इट इज डन, बट इज नॉट अ वेरी इजी टास्क because many types of cells will also become dead. So whichever cell will become zygote and divide to become zygote and blastocyst, that we will take and put into uterus of the female animal and then allow the fetus to grow, embryo to grow, right? And the birth of the animal will be the animal with all the chromosomes what have come from X, so will become identical. ठीक है, ऐसे ह्यूमन में भी हो सकता है. अगर मुझे अपना क्लोन करना होगा तो कुछ सिंपली यही करना है कि कोई भी बॉडी सेल मुझे देना है, ठीक है, किसी फीमेल से उसके ओवरी से एक सेल लेके उसका न्यूक्लियस हटा दिया जाएगा और जो मेरी बॉडी सेल का टोटल क्रोमोसोम है वो ट्रांसफर हो जाएगा और उसको जाइगोट बना के फिर उसको गिव बर्थ. तो एज अ चाइल्ड आई विल बी लाइक दैट, राइट. बट इज नॉट गारंटी दैट मूवी ऐसा नहीं होगा कि आई एम लाइक दिस सेम टू सेम एग्जैक्ट कॉपी. दैट विल नेवर प्रोड्यूस. अगेन इट हैज़ टू गिवन बर्थ, right? It has to grown up to that age, right? Then will look like me there, right? Development across lifetime that happens there. ऐसा होता है. हाँ ऐसा होता है. What you see in the movie के बस तुरंत क्लोन हो गया. Exact the producer is that technique has not come. That will take another 100 years, right? So what is the guarantee there? You can have that chromosomes there, but it's not guarantee that person will have the same behavior, right? Will resemble like me. कई बार development happens across lifespan, but the only thing is that have all the chromosome what I have. Okay, we can make Mahatma Gandhi, Nelson Mandela, but there is no guarantee there will be non-violent aspect of that, right? ये चीज़ है. Garima has been made like this. Our scientists in NDRI Karnal, they use this kind of, but our scientists are what? We don't get enough of fund. Our scientists do not get enough of fund. So what is called hand-guided cloning technique. UK में क्या होता है? ये सब के सब मैनिपुलेटर होता है. देयर लॉट ऑफ इंस्ट्रूमेंटेशंस आर देयर एंड दे आर एबल टू डू दैट. बट आवर साइंटिस्ट डोंट हैव सो सफिस्टिकेटेड लैब इंस्ट्रूमेंट्स. सो व्हाट आवर साइंटिस्ट डेवलप्ड हैंड गाइडेड विद द हैंड लाइक नाइफ एंड नीडल took the nucleus and put into the egg, that kind of transfer there. And desi ijad, there is a desi tarika, jugaad, what is called jugaad. So we have done that jugaad and we have made garima. So that is a kind of credit to the Indian scientists. With all those kind of local indigenous technique, we are able to make such kind of animal, right? That is what is happening there. So in the UPSC exam, the question which was asked, what is the technique used there? A, B, C, D. Name was given. What is the name? This one. This name was given. And this I have discussed in all the batches also, right? This is what you have to know. पता नहीं होता नहीं कब कहाँ कैसा क्वेश्चन आएगा. Whatever you are learning in all the subject is not my subject. History, geography, polity, economy, art and culture, what you read in newspaper, and then something is there in your subconscious mind, and the question comes. And this was direct question in the exam. कि how the cloning is done. क्यों पूछा मैं बताता हूँ. अब मैं current aspect पे आ रहा हूँ. 3 parents baby. What was making news? 3 parents baby. In the UK, first time a baby has been produced from 3 parents, right? Father and mother, the couple, and one from another female. And that technique also used a method of cloning. So instead of asking 3 parents baby, UPSC asks a simple thing, how the cloning is carried out. The name of technique. तो लोग क्या है, सारा 3 parents baby, सारा पढ़ते रहे, mitochondrial replacement, all this happened there, high level concept. But question which was very basic. यहाँ ये पूछ लिया गया, right? क्या किया गया 3 parents baby? Simple सी बात है. क्या problem था 3 parents baby में? क्या था? What was the, why it was there in the UK, such a baby was made? What was the problem there? Mother was, now till yet I have talked about only nucleus, but let's remember that. Mitochondria, which is called powerhouse of cell, right? They also contain, the mitochondria also contain nucleus, also contain DNA. Now let's say, for example, when we are talking with egg cell here, so egg cell will have the mitochondria. उसमें होगा ना mitochondria. Let's say, if you take any cell here, so one is the nucleus. Nucleus has got, let's say, for human being, how many chromosomes we have? 46 chromosomes are there. But we also have the here mitochondria, and sometimes mitochondria may be defective. There's sometimes genetic disorder can happen. There's a mitochondria which are there. Again, remember, then cloning also, the animal which will born will also inherit the mitochondria from the female animal, because that gene will also go there. But general aspect cloning, we only talk about nuclear DNA. We don't think in terms of this mitochondria. So इसमें क्या था? There was a defect. There was defective DNA was there in terms of the mitochondria. This was defective. So this defective DNA, let's say mitochondria was defective. तो क्या हो रहा था कि जितनी बार जो reproduction, fertilization, the child was born with the defective genetic disorder. So doctor thought of a way out. What the doctor thought of a way out? कि normal, the mother has this kind of mitochondria which is basically defective. So what was done was that a female was taken, right? इसको यहाँ पर क्या है? इसको fertilization कराया गया. Let's say this is egg cell. For example, अगर इसको egg cell कर दें, we take an egg cell here, so egg cell, right? And then you take here here sperm cell there. So what will happen there? Zygote will be made. Now zygote will have all the chromosome from mother and father. What will be the only problem? The problem will be that here mitochondria is defective. Defective mitochondria is there. Each is there. And so this is the actual parents, father and mother. But the problem is that this zygote which is made has got the defective mitochondria. Now what they did that they took another female. Another female was taken, and from female egg cell it also contains nucleus. This was removed, जैसे यहाँ पे remove किया. So now this female egg cell will be without the nucleus, but it will carry what? It will carry its mitochondria which will be functional, and this can be transferred. ये चीज़ है, same manipulation. बस केवल क्या प्रॉब्लम क्या था? जेनेटिक डिफेक्ट क्रोमोसोमल नहीं होकर माइटोकॉन्ड्रियल डिफेक्ट था. तो पहले साइंटिस्ट ने क्या किया? पहले एक को फर्टिलाइज कर दिया लैब में, ठीक है, जाइगोट बना दिया गया. अब इसमें जाइगोट में जो माइटोकॉन्ड्रिया है वो मदर से आया हुआ, एक्चुअल मदर से, तो ये डिफेक्टिव है. तो अगर ये चाइल्ड बॉर्न होगा तो इट विल बी हैविंग डिफेक्ट. तो उस केस में आइडिया क्या लगाया गया? एक फीमेल को एग लिया गया, क्योंकि उस फीमेल में जो माइटोकॉन्ड्रिया है वो नॉर्मल माइटोकॉन्ड्रिया है. हाँ, बस यही है कि उसके क्रोमोसोम नहीं चाहिए, उनका कोई जेनेटिक पार्ट नहीं चाहिए. तो दैट न्यूक्लियस वाज रिमूव्ड, पूरा न्यूक्लियस रिमूव हटा दिया गया. और ये जो यहाँ जो सेल का जो न्यूक्लियस बना है जाइगोट का, इसमें किसका क्रोमोसोम है? फादर एंड मदर का. तो दैट गॉट ट्रांसफर्ड हियर. एंड देन जस्ट लाइक क्लोनिंग, इट वाज अलाउड ट्रांसप्लांटेड ब्लास्टोसिस्ट एंड गिवन बर्थ टू द चाइल्ड will have all the chromosome of the father and mother, but will the mitochondria come from the female, from a healthy female, जो mitochondria. बस यही किया गया. तो UPSC asked the question because this fact was there in the newspaper. Instead of asking that, simply asked the method of cloning. So cloning is also involved here. ठीक है, तो cloning का बस यही होता है कि you have to transfer the nucleus. That is part there. That comes in. I'll give you a written part. You write there. I'll put in a paragraph. What I've drawn the diagram, I'll just put in a paragraph there. हाँ. मैं समझाऊंगा. अब एक बार लिख लो. I'll come again. Because biotechnology is totally biology based, so कई चीज़ें I'll explain again. I'll go slowly. I'll explain that so that everybody you understand. You can ask me doubt also there, question also, because ये सारी टेक्निक है, एक बार समझने के बाद things will be very clear. So now understand. Now write there. Write this. In reproductive cloning, somatic cell nuclear transfer technique. Reproductive cloning, somatic cell nuclear transfer technique is used. Somatic cell nuclear transfer technique is used to transfer nucleus from nucleus containing all the chromosomes, what I'm saying is nuclear transfer technique is used to transfer nucleus containing all the chromosomes into a denucleated egg cell, right? Give a full stop there. The egg cell is induced chemically. The egg cell is induced chemically to divide like a zygote. Is induced chemically to divide like a zygote to give rise to blastocyst. You have to stop there. This blastocyst is transplanted. This blastocyst is transplanted into the uterus. Of a female animal of a female animal where it grows into normal fetus, where it grows into normal fetus, FOETUS. So what is said? The nucleus, entire nucleus carrying all the chromosome, is being transferred to an egg cell without its nucleus. What is a denucleated egg cell? And the cell, what is formed, egg cell with the nucleus, is induced to become like a zygote and blastocyst. And that is transmitted back to any female animal. Remember that female animal, not necessarily that from where we have taken the ovum cell or egg cell. It can be any female animal, like a surrogate, and can allow the fetus to grow. So that will be the normal aspect which is done. I have given you a simple aspect, but in the lab there is a lot of manipulation is there. Okay. 3 parents baby was a mitochondrial replacement where the third cell was taken for replacement. That was done there. Right. Now what is done in therapeutic thing? I'll give you written part also. But let me first, before we go to therapeutic cloning, let me, हाँ मैं इसको समझा रहा था, वहाँ पर वो जगह नहीं था, मैं प्रॉपर समझा रहा था. 3 parents baby का ये किया गया था. Now this actually is what the manipulation, the different ways to manipulate, but I'll put across the general aspect which can be done. Now understand, ये सोचो आप, जैसे कि for example, the father, I'll explain with it, and let's say mother is there, right? Now father is normal, father have no defect. Let's say for example, the father here germ cell, reproductive cell, let's say here when it comes to sperm cell here, right? So it is a normal, it doesn't have, is a healthy individual, there is no problem there. But the mother egg cell here, egg and ovum, it was having a defective mitochondria. It was carrying defective mitochondria. Because mitochondria, remember that, also contain DNA, and sometimes genetic disorder, disease can be because of defective mitochondria. Right? Generally, normal cells में जो होता है वो chromosome का या genetic defect हो सकता है, but there can be also possibility. हाँ. नहीं, human body जैसे होता है वहाँ पर नहीं तो, but actually क्या है कि यहाँ पर जो egg cell है तो यहाँ पर just you have to screen it out, because what is saying that mitochondria defective is in the entire one or only egg cell, right? See, remember that whenever you talk about the cell here, the cell will have mitochondria there, right? But actual thing is that if you take a normal body cell, it will contain all the chromosomes. But what do you want? You want the father-mother component both, just like a fertilization happens. So here the mitochondria was in terms of the egg cell, the defective part. Because that's what, every time the baby which was born with a defective disorder and you died. You should die there. So what is the thing here, which I've said here? Let's say consider this is the egg cell, right, which has got here chromosome, here mother's one, so that will end half number. But the problem is that here the mitochondria which comes to that egg cell, let's say those mitochondria which comes, there is a defective mitochondria is there. Let's say for example this is defective mitochondria. Because if, remember that, if the fertilization takes place, let's say for example, if this fertilization takes place here, so what will happen? The zygote which will form, I'm talking normal situation. The normal situation when the zygote is formed, so here come n number of chromosomes, let's say for example, so here the zygote form, so what will happen with the zygote? It will also carry the defective mitochondria of mother. Normal situation, you carry the defective mitochondria of the mother also there. So that will, there the child will be born with defect. So this has to be corrected. It has to be corrected there. So what is the way out? The way out is that this mitochondria has to be replaced. But for that you cannot do with the mother egg cell. Because mother egg cell contains defective mitochondria. You require egg cell from another female which has got healthy mitochondria. So let's say third here, a female was taken. And from female this egg cell which was taken, so that was having here normal functioning mitochondria. Normal, it's a healthy mitochondria there. So mitochondria, but actually what is the thing here? You don't want that the chromosome of this female should become part of it. You want actual father-mothers to come there. So only thing is that the egg cell what we are taking here, this egg cell, the nucleus part, this nucleus, we don't want of that. This has to be get replaced. So what we can do, we can take it out. So when it is taken out, so what happens here? There is nucleus which becomes here. This egg will be there. It will have all the healthy mitochondria, but it doesn't have any nucleus. And what is the method here? We can transfer this nucleus. Because this nucleus already carries the half and half chromosome of father and mother, which can become part of this nucleus, egg. Right? Similar to what has been done in cloning also. Right? And this will become this egg cell. Now it carries all the characteristics of mother and father. At the same time, the healthy mitochondria, which comes from this female. Right? So mother's defective mitochondria doesn't become part of it. And then make it to divide like a zygote, and again as the cloning method does, same thing here. That no, this way it cannot treat it. What is asking that? Hemophilia, Down syndrome, this will not be, right? Because that is a normal kind of chromosome there, right? Which is part of chromosome defect. Down syndrome is what? Where an extra copy chromosome comes, that cannot defect. Here we are simply doing nuclear transfer, that's it. Right? If father or mother has any defect in chromosome or gene, for that you require other type of method. I will be coming to that method in today's class. That is something different is there. What is asking, something different is there. Now recently it has made all the news, and I'll tell you that this is very, very important because this has been asked in the exam. But next year you be assured with one question which can come CRISPR. But for that you want chromosome change or something, you will apply CRISPR technology there. Right? Here only mitochondria which was defective, that was replaced through using this method. That was done there. So उसके लिए अलग approach है. That is a very different approach. That is a separate thing. Down syndrome or hemophilia, which is there in the nucleus, nuclear chromosome is defective. So उसके लिए अलग approach चाहिए. And that has been now tried out with mutation in US. And China also, they're trying to treat cancer using the CRISPR technology, which is all making news. Whether it is ethical to do this kind of manipulation. Removal, cutting of DNA, and then adding the things.
Come under the cloning or?
Which one? No, this process, see this what I explained was cloning, but you can understand 3 parents baby is a modified cloning method. Just remember that. 3 parents baby is a method to produce healthy baby, right, using the cloning, modification of cloning technique only. But what he was asking that, let's say chromosomal defect or any other defect, we cannot use cloning. Cloning is just simply to transfer nucleus. That cannot be there, right? So that is a part there, because sometimes the question will be at conceptual level. The concept level you have to come. क्या पता हो सकता है this time mains exam question can be asked based on those kind of thing what is making the news, right? To whether remember that biotechnology as a subject is such a fast evolving subject and always comes with the ethical challenges and questions. And for that law is required. India is also struggling how we can go across those things which will become later in the society when such advanced technique will come for practice. So we are working on that, but generally remember that 3 parents baby is one of the exceptions which was tried out. That was the thing there, and that happened in UK. Scientists used, but actually the basic premise is the same as cloning. This I wanted to make because when you read from the current affair material, वो बहुत टेक्निकल आता है, प्रो न्यूक्लियर और क्या क्या लिखा होता है, तो द पीपल बिकम कंफ्यूज्ड कि क्या इसमें बायोलॉजी लिखा हुआ है कि हाउ द 3 पेरेंट्स. तो ये सिंपल अप्रोच है. दिस इज द डिफेक्टिव क्रोमोसोम माइटोकॉन्ड्रिया. You don't want this mitochondria to be there. You take this nucleus which is there in the zygote, which contain all the chromosome of father and mother, and then transfer to another female which has not got any chromosome of the nucleus but the healthy mitochondria in the zygote, then the egg cell, and that get transferred. That part is right. Now surrogacy. See, surrogacy only comes into existence when the actually mother is not able to receive, or in the uterus the zygote or blastocyst. Then you require the assistance of another female, and that female is called surrogate because in terms of using that uterus. Now surrogacy is 2 types, either commercial or altruistic. So surrogacy, commercial sense is called renting the womb. When you use the word renting the womb, that is in terms of commercial surrogacy. Why? Because that female is giving her uterus in return of money. That's called renting. But in India now we have banned that. But when it's an altruistic case, like sometimes sisters, maybe relative, close relative, may come forward to help. Surrogacy means simply is what? When mother is not there possible to take the zygote, take the blastocyst, we require another female to put that zygote there. That's where the surrogacy comes. Right? And two types, as I said, commercial or altruistic. When we look from money point of view, that's commercial one. So that is now banned in India. ART Act, Assisted Reproduction Technology Act, has banned commercial surrogacy because of all the issues and challenges that part comes. Right? Now I'll go to sequential. The first I explained the cloning, then I gave you the current aspect, how the 3 parents baby has been done, like how the cloning comes into the application when you're talking about 3 parents baby there, right? Now what I'm doing, I said in the classroom what, there's one is called therapeutic cloning, which I explained. But before we go to that, let's say, understand what are stem cells and what do you mean by stem cell therapy. The first step there. Then we'll go to the, again this part comes into that. So if we have to explain stem cells, so how we can put into stem cells there? Stem cells are they normal body cells? Like they're normal body cells. What type of cell? Can we say somatic cell or germ cell? There's a मान question aa jaye. For example, UPSC last time only came question, I'll tell you, only came question in stem cell was 2012 paper. After that, UPSC not in prelims exam, not in mains exam has asked anything on stem cells. Now what generally UPSC does when there's something more highlighted, like CRISPR was where he was highlighted. He didn't ask question. Stem cell is always highlighted in the newspaper. Again the question is not asked. So that's what, but again, any different way question can be asked. Like I have asked a question that when you say stem cell, are they normal type of body cells, somatic cell, or is it a like egg cell or ovum cell or sperm like germ cell? Where do you go there? Totipotent and all is there, but what type of, let's say body cells are, when you talk about humans or any animals, 2 types are there. है ना, 2 तरह का होता है. तो सोमैटिक सेल में ही आता है, जर्म सेल का उसका रोल नहीं है. मैंने इसलिए पूछा क्योंकि कई बार मैंने वो वर्ड यूज़ किया ना एम्ब्रोनिक, तो कई लोगों का एम्ब्रोनिक वर्ड है तो हो सकता है जर्म सेल हो. नो, दे नॉर्मल सेल्स ओनली which are present in the human body as well. I'll put across all the things, now you'll understand here. I'll go into that. Stem cells, first understand why they are called stem. Why it has not been given any other name. Why the word stem has come. This stem is not like tree or plant stem, root and stem. That part doesn't come. So here is that. First point is that I'm writing R. You can write also there. I'll keep on writing and reading out. R pluripotent, or let's write R unique body cells that are capable of dividing and differentiating into any other types of cells. They're capable of doing that. That means you can say they're programmed cells. They can be programmed. Right? So what I've given here, stem cells are unique body cells that are capable of dividing and differentiating into any other type of cells. Let's say, for example, this is stem cells, right? So stem cells can divide to form either the stem cells of its own type, right? Or it can get programmed. This can be programmed to become, let's say, kidney cell, right? Or it can become lung cells, or let's say retina cell. So you can produce any kind of body cells. Right, I'll explain this. What? Either they can form same type, similar type, or they can get programmed to form any other type of body cells. That's the capacity they have got. So we can say, since other type of body cells stem from them, arise from them, so they are named as stem cells. So in a simple word, we can say all types of cells arise or stem. That's why the name stem has come. They stem from them, arise from them. That's why called stem cells. Right? Now I'm coming to, they are basically, broadly speaking, a different classification is there, but broadly we can look at, we divide into 2 types, embryonic stem cell, adult stem cell. But sometimes in the books you'll come across pluripotent, totipotent, unipotent. I'll explain all of that one. But first of all, let me write the second point. There broadly are two types, either embryonic stage or they may be, let me write here, are two types. The first one is called embryonic stem cell. Right. Now, what are embryonic stem cells? Which are found in the blastocyst stage. I have already said are found in, I'll write here, are found in blastocyst stage of embryo. They are found there. They're found. And what is so unique about them? They are either pluripotent. I'm using the first one, pluripotent. Plural means multiple, plural type, right? And there's something called totipotent, total. Now there's difference there. That's what I'm writing here. Pluripotent means multiple type of cells they can produce. Can be generated. So what today we are made up of, all what we have got, right? Initially in the mother fetus has come from here. Because once the blastocyst formed, it divided and gave rise to everything which was part of our embryo development, our fetus development. So sab wahan se hai. So they are only in the embryonic stage, nowhere else. And when it's a totipotent, total type of, all types of cells. Like multiple type, I mean to say muscle cell, heart cell, kidney cell. But when you talk all the body cells, the total potential will come to that. But generally you'll find in the books, pluripotent word given. Right? Now what is coming here? Second type, adult stem cells. Right? Adult stem cells. That is found in individual, mature individual. Are found in individuals there. I'll give you one good example. Bone marrow stem cell. Right, in the bone marrow. These stem cells are very, very unique. They can only produce or generate blood cells. Bone marrow stem cell will not produce skin cell or brain cells or kidney or lung cells. They are very specific. They are programmed to only form the blood cells. Same way, brain stem cell will produce only brain cells. Skin stem cell will produce only skin cells. So they have what? They have got a special function. They have got specialized. So what we can write, sir, is right here, this thing. They are specialized, or what we call unipotent. I'm writing here, they are unipotent. So that is what? Specialized type of cells. They can form specialized types of body cells. Like for example, I write here, for example, bone marrow stem cell. Like bone marrow stem cells can only produce blood cells. But when you say embryonic stem cell, the embryonic stem cell can form other cells also. That's what capacity it has got. It's basic fundamental concept. That's you are aware also there. I'm just trying to put in a point wise from here. So there's a brain stem cell, brain will produce only brain cells, not any other type of cells. So they are very specific. Right? They are very, very specific. Okay? Now let's say, for example, these things are there. Now, you will also be coming across, I am very sure you are aware, there is something called umbilical cord blood, right, which is placenta. So it also contains stem cells, right? And that happens in the placental stage, placental stem cells. And when a child is born, there is the umbilical cord that also contains there, right? But I'll tell you.
The cells produced from the stem cells all are equal or different from each other?
No, what does that say? When you take this type of cell, embryonic stem cells. Let's say, for example, I'll explain here. What is the last question? Let's say bone marrow stem cell. The bone marrow stem cell can divide to form bone marrow stem cell. Same type as the cell divide. Like in our body, what happens? Skin cell also divide. All the body cells also divide, and they form the same type. So let's say bone marrow stem cell can divide and form bone marrow stem cell. or they will get programmed to form WBC, white blood cells. So that is blood cells. Actually, it is a stem cell, but now giving rise to white blood cells. WBC is formed. So that means they have got a function to produce a WBC. Right? So they can form either bone marrow stem cell of own type, or they can divide. And what I've used about differentiation with a function to produce WBC, white blood cells. Right? Let's say brain stem cell, brain will divide to form brain stem cell. At the same time, they can form other kind of neurons, motor neurons and all this. So sometimes what happens, we get damaged also, so that get repaired. So those kind of cells can be replenished. Right? Now why it has got so much of attention? Because of the capacity to give birth to cells. So what is something called regeneration or regenerative medicine. Right? So what I am coming here, if you look at the application part, so their applications, what comes here is called regenerative medicine. So since they can regenerate cells, so we can use for number of therapies. So I'll give you the areas of work which is done today worldwide. The third point is that I'll come back to umbilical cord also there, but let me first write application there. The application part here, what comes here, what I'm writing here is called regenerative medicine. This is a branch of medical science, regenerative medicine. That means we can repair any damaged tissues and cells by the use of stem cell. So that is the repair, what I'm writing here, or repair of damaged cells using stem cells. They are also sometimes called as tissue engineering. Right? Now normally if you look at their part which comes, so we can go for cancer treatment. Cancer is one, cancer therapy can be done using stem cell. Like for example, I'll give you example, treatment of blood cancer. Right, blood cancer. Now why blood cancer happens? When bone marrow, right, is affected. So what happens? The WBC and all these are not being formed. So this blood cancer, so what is done in normal case in the case of hospital? Bone marrow transplant of blood, right? A person's bone marrow has to be transplanted from another person, they get transplantation. But sometimes what happens? Graft can be rejected. तो वहाँ पे क्या चाहिए? जेनेटिक मैच चाहिए और बहुत ही डिफिकल्ट होता है कि बोन मैरो ट्रांसप्लांट कराना. तो आज हमारे पास क्या ऑप्शन है कि हम बोन मैरो ट्रांसप्लांट नहीं करा के जो डिफेक्टिव बोन मैरो है उसको हम बोन मैरो स्टेम सेल से ठीक कर दें. स्टेम सेल कम्स इनटू दैट एंड टुडे इज बीइंग डन. देन आई विल राइट हियर व्हाट rare blood disorder, you can write this also there. Therapy like rare blood disorder. Sometimes we can think of, let's say, person who suffers thalassemia or some kind of disease there. So that sickle cell disorder, this all can be treated also there. Rare blood disorders. I'll give you case study in Chennai. Right, the first child was born, had a problem with this kind of thing. Right, from the birth the problem was there. The doctor advised that when the second child is born, to remove the umbilical cord blood stem cell and use for the treatment of first child. Why it can be used? Because in the same family, the genetic match will be there. Right? That can be done. So that was done, and then successfully the first child was treated with the disease, which was done in Apollo Hospital. Today is being done worldwide. This is one of the things. Lisa Ray, the film actress and model she was suffering with blood cancer, but now she has recovered because of stem cell therapy. So worldwide this is being done. Now another thing is that treatment of blindness. Right? So blindness can also be treated. Like why blindness happens? When retina is damaged. Then only we will not be able to see. So if we can repair the retina with a normal retinal cell, so that can be there. Again, the worldwide there has been attempt for treatment of blindness treatment there. But actually the thing is that when you talk retina and all, it requires again very much a lot of manipulation things there. Cancer case is very much is there. Like so, any defective organ or any defect there, we can go for this stem cell. Through stem cell, we can replace those defective cells. Right? It is said that in future, with advancement of stem cell therapy, one can grow the organ in the lab. And again, it has been tried out worldwide. Like blood vessel has been regrown, right? Even using 3D printer, organ has been now being printed using stem cell there. That is a what advanced type of stem cell research. In future this will happen. Today it is not possible that we can grow kidney or lungs or heart and go for transplantation. Maybe future can happen. Now what I said is the fourth point. Let me come to that. So I'll come to fourth point is that. Fourth point I said here, stem cell banking. Stem cell banking, that's what I said here, storage of umbilical cord blood. Umbilical cord blood stem cell is done there. That is done in the lab and then kept in a preserved case, right, at low temperature. It's kept for a long time. Again, the question is raised that whether cells will be really viable after 20 years, 25 years. Will not the cells damage? How then can we use? Let's say child is born and the parents go for stem cell banking. And when child grows to adulthood, 25 years and requires stem cell therapy. And at that time stem cell has not been there. So again, there are problems. But actually, those companies which there, they have a responsibility to do that. So यहाँ पे कई बार insurance वगैरह भी आता है. तो ऐसे कई company है who charges money for that. You have to pay money for keeping your stem cell of your child preserved for a long time there. That's what. And this is what is happening worldwide, including India. Lekin, there's something miracle has happened. What is miracle has happened? There's one advent of technique called iPSC. Like our parents have not stored our umbilical cord blood. And let's say we want therapy in future. So again the stem cell gives an idea because stem cell का सबसे ज़्यादा utility कहाँ आता है? Embryonic वाले यही है ना? सबसे ज़्यादा fruitful क्या होगा? ये होगा. Very important. Embryo. कहाँ से मिलेगा? Embryo से. And taking from embryo will be not advisable. Why? It will damage the embryo. That's the only thing. From where embryonic stem cells are? The blastocyst. And you want to take from blastocyst, you have to destroy the blastocyst. That is really not good. It is unethical. So for that reason, embryonic stem cell was banned. Research was banned. Right? So there came a miracle. A Japanese scientist, Shinya Yamanaka, he devised a method in the lab where one need not have to take embryonic stem cell and still can get the therapy. Right? And he was awarded Nobel Prize. Nobel Prize in Medicine Physiology has been given for his work, what is called iPSC, induced pluripotent stem cell. So now I'm coming to that point. So this fourth point of stem cell banking. But let's say we have, our parents have not done, but we want therapy. So how we can go for? So there's another method by which stem cell can also be produced. So I'm now coming to that method. What exactly iPSC is doing. So what is the next point? Fifth point is that induced pluripotent stem cell is a called induced because ये वर्ड काफ़ी बार न्यूज़पेपर में आएगा. बार जब आप पढ़ेंगे हिंदू इंडियन एक्सप्रेस, all these technical words will come. I will use those kind of, even sometimes abbreviation will be given. And at that time, if you don't have the background, you will not be able to understand what sense it is talking about that research. So that part, induced pluripotent stem cells, iPSC. Now what happens in this case is that, I'm writing at the point where normal body cells can be induced to produce embryonic stem cell. Remember that they are not actually embryonic. We are inducing to make it embryonic stem cell. Embryonic stem cells through what I'm writing here. There's something called gene through insertion of few specific genes. What these genes are there, first time was worked out by Shinya Yamanaka. Right, first time, what genes will be there, which will be added. First time it was figured out in the lab. Right, it was done in the lab by, now I'm going to this name, this Japanese scientist, Shinya Yamanaka. He is a part of Kyoto University. There is a chair on, there is a division on stem cell research, Kyoto University in Japan. So Dr. Shinya Yamanaka, he first time in the lab he came out this particular type of genes. He figured out certain type of genetic sequences which when added to normal body cells, let's say skin cell, the skin cell becomes stem cell. And these genes are also called Yamanaka genes. They're named after the scientist who first time found out this particular genes. तो उनका नाम भी दिया गया यामानाका जीन्स बोला जाता है, क्योंकि उन्होंने फर्स्ट टाइम लैब में ये फिगर किया कि अगर ये जेनेटिक सीक्वेंस को ऐड कर दिया जाए तो नॉर्मल सेल को हम स्टेम सेल बना सकते हैं. That's what comes here. So what happens here? Now this embryonic stem cell produced, and from there you program to make any other type of cells. Right. Now what is the second thing here? These induced pluripotent stem cell can be programmed. Second point you can write, can be programmed into any other body cells. I'll explain with a diagram also there, any other type of body cells there. Let's say for example, if I take simply skin cell and then we induce by adding this the 4 type of genes which are called Yamanaka genes, right? Whatever sequence will be there, the genetic manipulation, and this skin cell becomes like an embryonic stem cell, pluripotent stem cell. It gets converted. And then from here, whatever we want to make kidney, we can go for kidney cells, or lung cells, or heart cells, whatever we want to make, let's say, we can make it there. So that is the indirect way. What is the indirect way? So it is possible that in future, we can also get treatment. Simply scientists will take any body cells, do this manipulation in the lab, induce to become a stem cell, and from there make any type of cell. In Japan, it has been tried out. In Japan, a lady was cured of blindness, retinal cell, and that retina was developed in the lab through this method. ठीक है, लेकिन इससे प्रॉब्लम क्या है? What is the problem in this case? Did you understand? जैसे अब स्किन सेल लिया गया, अब उसको हम लैब में IPSC बना दें और फिर वहाँ से रेटिना सेल बना दें और फिर वो रेटिना सेल को हम ट्रांसप्लांट करा दें. ये मेथड है, लेकिन इसका प्रॉब्लम है. क्या प्रॉब्लम है? It's today is not very full proof there, but what problem will be there? What about this retina cell? Let's say अब आप समझो आप retina cell आपने बनाया, and what this retina cell understand that it is not retina cell but the skin cell. This is what you have done. Memory can come back. It's possible. ये क्या है? It's like hitting the head with a hammer. किसी सेल का अगर मेमोरी चला जाए तो वो स्टेम सेल हो गया. Because stem cell is what? Without any function. Right? So let's say you made that and retina cell returns back to the skin cell. So what will happen? Big problem. आपने तो स्किन सेल के लेकर रेटिना बना दिया, and if the retina cell returns back, goes back, so that person will again suffer a lot. So that's what it is. Today it is not full proof. Scientists are working. What second problem could be there? This kind of cell will also die. They will not have long longevity. या ऐसा हो सकता है कि आपने क्या किया, फॉर एग्जाम्पल ये हार्ट सेल बनाया और हार्ट सेल व्हाट हैपन वेरी फास्ट मल्टीप्लाई लाइक कैंसर सेल, अगेन विल क्रिएट अ प्रॉब्लम. तो साइंटिस्ट आर वर्किंग. Yamanaka, Dr. Yamanaka did only in what? Mouse. He did this work with chuha, not with humans. So with humans it will take time. Right? Scientists are working on that. It will take time. But he just only tried in the lab with a mouse cell. He proved that a mouse cell, skin cell can become stem cell and can produce any other cells. But in humans we still have to work out. We are working that. Okay? This is what comes into that. So 2012 Nobel Prize in Medicine. He got 2012. Nobel Prize was awarded to him. Half of the prize went to him. This ये केवल लैब में जनरेट करते हैं. In between the temporary generator, and then made into the any kind of cell. ये चीज़ है. So that's what I said. In future, a lot of things can happen in stem cells. That's what scientists are working on that. Okay? But this is kind of basic consideration with that. Half the prize in the 2012 went to Dr. Yamanaka, and half the prize was given to a scientist. I'll give there that general knowledge also there. Dr. John Gooden. And now I'll tell you that Dr. John Gooden is said to be father of somatic cell nuclear transfer. You can understand, 2012 was known for cloning and stem cell. Together was recognized by Nobel Committee. So Dr. John Gooden is a UK scientist. He developed this process of somatic cell nuclear transfer. But he didn't try with the mammals or this one, but he tried with the tadpole, frog. First person to carry out in mammal was Dr. Ian Wilmot. In 1996, Dolly was made. Right? That was the first time in mammal I'm talking about. Dolly is a ship there, the bhed jo tha. But actually, in way back in the 60s, Dr. John Gooden, he showed in the lab that nuclear can be transferred. But he did in the frog, right, which later on was tried out with the other kind of animal system there. That part. Dolly ke case mein, almost around more than 500 ships were sacrificed, right, killed, because all will not become successful, only one will become successful. So that also gives rise to ethical question. If for the sake of cloning, should we kill those kind of animals only to get this? There's also ethical questions have been raised. Again, Dolly didn't survive for long. Dolly also died. Why it will die? Very simple thing. All we are doing in what? Lab. We are making all this artificial zygote. Artificial thing. So this will be not a natural zygote. The zygote which we make in cloning through also is not natural. And that can return back. So बस basic concept को ध्यान रखना कि what I'm writing key point. This is enough. Right? If you keep this point, any question comes in the exam, you'll be able to get to that. Right? Now I'll open a particular thing and I'll show you one documentary, a very unique one, very interesting one, and you'll get to understand exactly what has been done. Right? So ये दो technique which I've come across there. Now therapy case में यही होता है कि which will I keep on moving a lot, right? And I write a lot of things there. So I don't know people at that side will be able to see or this side will be able to see. So if you want me to restrict to a particular place, then I'll restrict there only. It's all right. You can go through this one. थोड़ा मेरा वजन भी कम हो जाता ना. When I go from left to right, so whatever I've eaten, so that also get digested. एक digestion हो जाता है वहाँ पर. आज के टाइम में बायोटेक्नोलॉजी कैन डू एनीथिंग, दैट इज ऑल पॉसिबल देयर, राइट? सो सुबह सुबह उठते अगर आपको लगे कि नहीं, आई एम नॉट वर्थ इट, आई वांट माय बेबी टू बी ही मैन, सुपरमैन, हनुमान, दैट इज आल्सो पॉसिबल देयर. दिस ऑल क्रिस्पर एंड ऑल हैज कम, राइट? दिस व्हाट हैज बीन डन देयर. आज लगता है इंक बहुत अच्छा डाला है. इज ऑल इज नॉट गेटिंग बिकॉज द You're not going there. ये ऑन कर देना. There is a iris stem cell bank, iris research lab on stem cells. I am playing that video only, and you'll understand the things there. Come there. Play it.
This is a short movie by the Irish Stem Cell Foundation, which seeks to educate people about what stem cells are and what they can do for us. The human body is composed of over 200 cell types, each designated to a particular function. So, for example, a pancreatic cell is involved in the function of the pancreas, and the nerve cell is involved in the function of either the brain or spinal cord. A subset of cells, however, are different. These are called stem cells. Stem cells have the capacity not only to make more stem cells, but also to make diabetes or or say motor neuron disease. There, the problem is that there doesn't appear to be an adult stem cell that can make insulin secreting cells of the pancreas, for example, or like motor neurons that are lost in motor neuron disease. Scientists and clinicians, in order to find out what's going wrong and to fix what's going wrong, they need the type of stem cell that can generate the cell type relevant to the disease. So in certain instances, embryonic stem cells are needed. People frequently think about how stem cells can be used directly to treat disease. For example, for many decades, bone marrow and the stem cells they contain, blood stem cells, have been used in the treatment of certain cancers and rare blood diseases. Scientists and clinicians today are even more excited about the indirect use of stem cells as research tools. We can learn more about how stem cells make different cell types. We can use cells derived from stem cells to make better and more effective drugs. So stem cells are, in essence, a source of different cell types that you can use to generate new drugs and learn more about how disease happens. You know, it's frequently said in the media that embryonic stem cells are no longer required because of the advent of a new technology called induced pluripotent transduction. And this is simply not true, because embryonic stem cells will always be needed to study early human development and pathologies and diseases associated with those early stages of development. Forget that they're a particular type of person and they have to relearn what kind of person they are again. But sometimes memories re-emerge, and this is really where the state of the research is now. Can we make a cell completely forget what it was? And can you sustain that amnesia long term within the cell? This new induced pluripotency transduction is a very, very new technology, and we're still trying to find out, how we can make cells forget what they are so that they revert back to this stem cell-like state. But really, it's too early to say for a lot of applications whether embryonic stem cells or induced pluripotent stem cells will be better in future. The Irish Stem Cell Foundation is basically a collection of doctors, scientists, patient advocates, patients, and policymakers that are joined together by their expertise in some facet of stem cell biology, either looking at how these cells can be used therapeutically, how they can be used as a research tool, what kind of ethics do we need in order to make sure that good research proceeds. We also need to understand the needs of patients, and we also need to understand the consequences of not having good legislation pertaining to this area, and how that affects not only the science and medicine in this country, but more importantly, the patients in this country.
Ireland, it is a case of every country. Let's say when stem cell therapy will become more common in India, we also require a law for that. So now recently 2 laws have been coming in the news. I'll be now coming to next technique. See, therapeutic cloning is what? For therapy. We make a blastocyst, take the stem cell and go for therapy. But let's say there's one recently, a law has to be passed by the parliament where a bill has been introduced. DNA profiling. It's all again made, Hindu paper has come. The DNA profiling test. Today we are doing in the country for DNA profiling. So now since let's say, in the bill what happens? In the bill there is provision for regulating that kind of areas. What are the issues and concern and how DNA fingerprinting can be done. Different aspect of that. So let's say I'm starting from there. When you look at here, in this case, DNA fingerprinting and all this. But first I'll go to this. There's a one thing which I've said in the classroom, but again I'll write here. There's a Harvard University project on revival of woolly mammoth. Revival, and it's not only scientists are trying to revive woolly mammoth, but they're also trying to revive other type of extinct species also. Now this woolly mammoth revival, it's called Revive and Restore project. So I'm adding just from the current aspect that there's worldwide lot of attempts are being tried out from biotechnology perspective, and that comes into conservation part. Even we are also doing in the country. I'll be taking to the particular aspect this part. Okay. Project Woolly Mammoth. So once woolly mammoth, there's a friendly mammoth, right? Used to roam around in tundra habitat, Arctic and all this part, and then they become extinct with change of the season, geological time span. Now, what the scientists want to recreate the mammoth back, as it happened, you have seen in the movies there, and they're going to use two methods, cloning and CRISPR, to edit this particular thing and make a. Because why CRISPR will come here? CRISPR will only come because today original mammoth is not existing. Even we cannot have egg or ovum from mammoth because there is no female mammoth. So which is the nearest one? Elephant. So in that case, what has to be done from the fossil, whatever chromosomes are there, has to be reconstructed with the help of elephant chromosome. So for that we require CRISPR. What is CRISPR? Gene editing tool, which can cut and join together. So what Harvard University scientists are doing, they are using CRISPR technique to rebuild the entire map of a woolly mammoth, and that is done with the help of Asian elephant. That's what is being. And what they will do? Then they will do the cloning. They'll carry out cloning, and the elephant will give birth to the woolly mammoth, which will not be exactly true mammoth, but will be modified elephant and mammoth. This is what they are planning to put into tundra habitat. Now this here, when you look at here, this particular part, woolly mammoth there.
It goes wrong.
No, that's why ethical questions are raised. Some questions are raised. If you are trying to do that, but if something goes wrong, so who will be responsible for that? Is there any need to do this? Then again a question comes. Is there any need to invest money to develop this kind of animals? Now I'll tell you what is the positive outcome of this project. Positive outcome of the project is not to go to ice age, return back to ice age. The aim is that recently extinct species, let's say 50 years back, which have become extinct, at least we can try out this kind of method to bring them back. That instead of going millions of years back. That is what the immediate benefits are there. But again, the problem is that we have to regulate also there. So now this here, this woolly mammoth project here, the true story. Now what is said here? The possibility of biotechnology. Project goal. Now I'll come to this. The project goal. I'll just read it out. Breakthrough advances in genomic biotechnology are presenting the possibility of bringing back long extinct species, or at least proxy. What is a proxy? Which will not be exact. Right, proxy species with traits and ecological functions similar to the extinct originals. Similarity, what is being said. And woolly mammoth has emerged as the leading candidate for this work. Right, that is what it can be attempted because a close relative of mammoth elephant exists. That's why the scientists have gone for mammoth. Dinosaur we cannot do. Why? We don't have any close relative of dinosaur. If we can go for cloning there, right? But elephant we have, so we can try out this one. This is what is said here. And it said here Eurasia and North America, where this animal was frequently found, but now no more found. So after making this mammoth, they will put into tundra habitat. That's what they are planning there. So maybe in future, if they are successful, so there may be tourism, people may go to see how the elephant. But actual objective is that adaptation of Asian elephant with cold climate. How they can adapt to that condition. That's what is point of. So there is a why bring back and all this, given there is de-extinction.
So can't they collect the genes from the fossil
evidence? They're from fossils only. DNA will from, because they only intact, there is no intact DNA. There is no cell, you can take only from fossil. That's it. Now look at here. एक और current aspect में news आया हुआ है, I'm adding that. And let's say the question comes in the exam, current aspect, I'm adding this. Our scientists can also do, Indian scientists have also trying to go into biotechnology, and there's something called lacons. Hyderabad. What we have done in India, we have developed a lab, and lacons stands for, let me write here, the lacons stands for lab for conservation of endangered species. It is given in the Science Reporter also. This time Science Reporter June month is on the wildlife, and you will find this kind of things are mentioned here. So in India, in Hyderabad, CCMB, Center for Cellular and Molecular Biology, CCMB campus in Hyderabad, which is a autonomous campus, autonomous center of DBT, Department of Biotechnology. This particular lab has been established. It has been now established here, Laboratory for Conservation of Endangered Species. So even in India, we can try to bring multiply tigers, elephant, or any extinct species. We can bring it back. There is a center for this one, Center for Cellular and Molecular Biology. Right, this is a funded lab of Department of Biotechnology. The lab is there, and within that lab, we have now set up a this kind of lab there. Lacons is there, right? Now, if you can look at here about Lacons there, I'll just open this website because this is again remember that this Lacons and all have come in the newspaper for last 6-7 years, but not regularly, occasionally, right? So again, I'm going to that topic so that if in case UPSC asks anything direct question next year, so at least you're aware that something is there in the part of it. So what I want to bring, if you have already environment class, but I'm trying to link there. When you talk about biodiversity conservation, there's something called in situ and ex situ. So this is ex situ conservation. We can use biotechnology technique to revive the species or increase the numbers. There we can use the biotechnology part. Now this is what you have the lab there. What you see the infrastructure and all, this part is given there. And here you'll find CCMB written also. Below it is given Center for Cellular and Molecular Biology. And here if you look at achievements, अभी जो news में आया हुआ है, ये achievement है. क्या लिखा हुआ है? Development of universal DNA marker, DNA based marker for identification of wild animals. And the derivatives, like sometimes क्या होता है? Illegal trade. Sample specimens are there. So how can we get to understand that this is sample or skin of Tendua, leopard, or coming from panther? So we can identify through DNA which animal it belongs. Again, that is part of wildlife crime, right? DNA sampling and all. You know, census. You have come across cow, this dung, elephant dung method. So what is dung method? Dung method is that from elephant feces, right? Again from DNA, trying to assess which elephant is there. That is done for tiger census also. So now see, I am adding biotechnology with the environmental perspective. Okay, how the technique can come from DNA, we can find out which tiger, which elephant in the forest. Again, that method is used, right? Tissue culture, you know, tissue culture plant. We can regrow the plant, same type, clonal population. Again, that is a method of propagation, at the same time conservation. So this is all they are trying this. Now, this is what you see here. DNA banking of more than 250 species of mammals, birds and reptiles in India. This has already been completed. This particular thing. Even for the main exam, when you write something mixed type of question comes, you can add that. Right? So their DNA bank is all having sequence information there. This is what is given here, right? And this, I think this one is smuggled. Now see second point, rehabilitation of smuggled star tortoises based on molecular marker studies to their native range. Star tortoise you will find in Chinar Wildlife Sanctuary. There is Idukki in Kerala. There you will find this particular type of tortoise. And their research and rehabilitation again, DNA has been used. I'll give you one more example, butterflies. For butterflies, DNA studies has been done. Again, in our country, such kind of approach has also been developed. What has been done worldwide? Clear? Now, as I said, what? Now, recently is making the news. I'll first open that law there, and then I'll come to technique part. I'll be coming to technique part of this. They just I have given so that you can go to this website and can see how the environmental conservation is coming from the biotechnology application. That part is there. Now, if you can look at here, what I want to say here, DNA profiling test, this bill, which has come in the newspaper, bill, this act. So you all know that whenever you have to read about bill and all, there is a website called PRS. The review system is there. And all this, what has gone to Lok Sabha, Rajya Sabha, whether enacted or amendment has been done or pending bill, all you'll find in the PRS website. Now this is the copy which has been there in the PRS website here. Now see what is given. I'll just zoom out there. Let me just. Anything which comes in the current news with the basic concept is always related. Now this is different. to regulate the use of deoxyribose nucleic acid DNA, analysis of human body substances, profiles, and to establish the DNA profiling board. The law is talking about, this has not been passed yet, but has been tabled. But it's saying that there has to be DNA profiling board for laying down the standards for laboratories, collection of human body samples, substances, custody trail from collection to reporting, and also to establish a national DNA data bank. The word is given national DNA data bank of the people for which the DNA fingerprinting will be done. Right? And from matter connected therewith or incidental to this. This is a human DNA profiling test. Now, why it is necessary for the law? Let's say sometimes there might be possibility of misuse also. Something can happen because, again, you know the privacy of the data. Let's say your sample, DNA information, if it gets something outside and somebody gets to know the entire thing, the confidentiality, all this comes, and legal matter, particularly the case of criminal cases, all this become very important. So in India, we don't have any strong privacy law where we keep it, but in UK, European countries, they are very, very much obsessed with the privacy part, and they have very strong law in the country to regulate all this one. Right? So I'll not remove, I'll keep it there. So this is what objective of the new bill is there. ठीक है? अब इसमें क्या यहाँ पर है? ये तो हो गया current news. What is this current news? What you have to come? You have to come to the basic biotechnology technique. What exactly DNA profiling is all about, how it is done there. So I'm now coming to next technique called DNA fingerprinting technique. Anyone in the classroom who can tell me what exactly is done in the lab? Or this kind of technique there? And again, I'll point wise, I'll put everything on the board there, which you have to know from the UPSC exam point of view. Yes, anyone who can throw light, Prakash daliye, any light on this particular DNA fingerprinting?
DNA fingerprinting, the mother sample
and that father mother is all right, sample there, but exactly what is done in the lab? Here I'm coming to technique part. Okay, what is the technique? What is done in the lab exactly? Okay, all right. Anyone from the biology background, biotechnology background wants to know about, tell me that what exactly this technique is done in the lab? Again, what I said, from the UPSC, remember that, from UPSC point of view, you have to what you have to do is that just know there the terms and the meaning. Let's say this bill is making all the news. So it can be the question asked in the PT exam, something on that. Right, 2 or 3 statement type question there. So again, the very basic understanding, now I'll tell you that. In 1984, right, a UK scientist, Dr. Alex Jeffreys, he was working in the lab and he came out to a certain piece of DNA which is unique to an individual. So what I'm writing here, first point is that is based on, you can write a note copy, is based on, I'm writing a variable number of tandem repeats. What I'm writing the name of sequence there, variable number of tandem repeats. एक तरह का ये repeats DNA होते हैं, which is randomly repeated in the chromosomes, right? And these are sometimes also called microsatellite DNA. I'll write on the board also there. V and TRs. They act like a fingerprint for finding out the person, right? Now these are they are the part of something called microsatellite DNA. अब ये समझो कि जितने DNA होते हैं, ठीक है, हमारी बॉडी में, all are not gene. What is gene? Which has got trait, feature, character. Right? Human body has got 46 chromosomes. We have got 30,000 plus genes, but still we have DNA which has no function. तो कुछ ऐसे DNA भी हैं जिसका एक function हो सकता है fingerprint. That was called variably numbered tandem repeat. I just give example, very simple example. Just take example. I write here AATTCC. How many times this has been repeated? Maybe 1,000 times, 10,000 times, 20,000 times. Same way, let's say TTAA. How many times this is coming? 6 to 8 pairs. So that will have number of permutation combination, and that will become uniqueness to the person. That's what said. First time in the lab, such sequence was discovered by a scientist, Dr. Alex Jeffreys. He is said to be father of DNA fingerprint. So this is what comes second here. This DNA which is found there, Which one? Ajay, I'll tell you. This is a certain type of DNA, tandem repeats. You can say this is a DNA part is there. And which are unique to a person, unique to an individual. Okay, how they are found in the person. Let's say my VNTR will be only mine. Will differ from you, differ from him. Like a fingerprint happens there. So first time this was sequence of DNA was discovered in the lab by Dr. Alex Jeffrey. So I'll write this. But discovered this, developed. This method was developed by Dr. Alex Jeffries. He came across that human body. He came across in the lab that there's certain type of sequence, right, which can act like fingerprint, which can be unique to the person. And this is the name of the sequence is VNTR, variable number of tandem repeats. That is called there. So they came across this part of that, right? And what is done here? Now what I'm writing here, there is a molecular biology method, and so the gel electrophoresis bolte hain. So let me here. DNA band pattern, you can write DNA band pattern developed on a gel electrophoresis. I'm writing in the bold form there, gel electrophoresis. So ये method है, this is a technique done in the lab, right? So what is done in gel electrophoresis? I'll change the marker there, so I'll make it more black, so you can read there because there is already lighting also. Gel electrophoresis. What happens? Gel electrophoresis. In the lab there is a device. ठीक है, वहाँ पे क्या करते हैं? इस तरह से है. Let me. This is kind of machine or instrument is there, this type, and the medium is here agarose gel, semi-solid gel is there, right? Let's say for example, it is connected to the power supply. There's the power supply, it is connected there. ठीक है, अब example में देता हूँ. For example, spot of crime where murder was committed. So what will forensic team will do? Will collect the sample. They look for any kind of living sample, let's say. And in the lab, what they'll do? In the lab, the test tube, they will break these cells and will get all this DNA here. So let's say this spot of crime, where the on the spot forensic team obtains some sample. So what they'll do? They do in this gel electrophoresis. This is all DNA, pieces of DNA. They will be of different size. उसका length अलग-अलग होगा. So they add on this one and they add this sample here. And they run on the gel there. So just like paper chromatography, paper chromatography, जहाँ पर the colors get separated based on size. So when you apply current, so DNA is negatively charged, and they add certain type of dye, fluorescent dye or something. So what happens? This will get separated because you know that longer piece of DNA, longer DNA will be heavier, will move slowly, and a small piece of DNA will move faster. So they will all get separated. What is called band pattern generated. Now let's say suspect number 1. You have suspect number 1, suspect number 2. Now let's say sample is taken from there. So what sample can be? Saliva, right? Skin, anything where DNA can be obtained. And in the lab they will run this one. This will run in the lab. And let's say suspect number 1 is the culprit, for example. So what will happen? This DNA and this DNA will match. If suspect number 2 is not a criminal, so the band pattern will not match there. So that becomes a kind of conclusive evidence that the person was there on the spot of crime. That's the sample matching is there. क्योंकि ये जो sample वहाँ से है जहाँ crime हुआ है, और दूसरा यहाँ से उसका match कर रहा है. So that means the identical matching is there. DNA is coming from the same. तो ये जो DNA है, these are called VNTRs, which are separated in the lab and run on the gel electrophoresis. So for UPSC exam, again I just remember that you don't have to go to the lab protocol, okay, how the separation, how the DNA cut and done in that case. But simply remember that there is a device called gel electrophoresis device, and the DNAs are sequenced, different DNAs are separated. And what the scientists do, they take a photograph. They take a photograph of this pattern, and that is submitted as evidence in the court of law. Okay, this is the DNA fingerprint, which the matching is being done, right? And that becomes a kind of in the trial gets done. Now, since this technique has become very popular, many countries are trying out this method of DNA fingerprinting to nab the culprit. This has come into that. So you can write the next point there. I'll just after this you can write. DNA fingerprinting. DNA fingerprint you write. DNA fingerprint is obtained on 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 a gel electrophoresis device, on a gel electrophoresis device in the form of on the gel electrophoresis device in the form of DNA band pattern, in the form of a DNA band pattern. DNA band pattern. Okay, first of the suspect samples from suspects, right? Samples from suspects are also run. Samples from suspects are also run. Are also run on. Samples from suspects are also run on gel electrophoresis, and DNA band patterns are compared. Right? Now last sentence after DNA band patterns are compared. Now write this. If DNA band pattern of a suspect, if DNA band pattern of a suspect matches with the DNA band pattern of sample DNA band pattern of sample obtained from spot of crime, comma, it becomes conclusive evidence. Of the person's person apostrophe s person's involvement. It becomes a conclusive evidence of the person apostrophe s involvement in the crime. Person's involvement in the crime. Right? It becomes a conclusive proof that the person is there in the place there. But remember that if somebody has changed the sample, there's something there. But if the sample is without any tampering, no tampering has been done, and that comes there, that becomes a. Now I'll come to that. Paternity and maternity cases are different. Why different will be there? This happens for the normal individual identification. There what will happen is that you require father sample and mother sample. So let's say in this case I'll just change it. Let's say son or daughter. Your son or daughter, the sample is taken. So it will generate the band pattern here. So 50% match will come from father and 50% from the mother. Right. I'll just give an example. I'll show you one, and let me ask that if you can tell me that whether this is the father or mother or not. Let me just. I'll just show you one band pattern, one case there, and tell me that whether the person will be the father of that particular child or not. Now this is Dr. Alex Jeffrey. 1985, he came out with all, and then now see here. This is what happens. This is gel where the band patterns are separated, right? And they can be visualized. Sometimes radio labeling, this kind of will be there. Now look at here. What type of samples can be taken? So you can write this. Biological materials, you write this. Biological materials used for DNA profiling. Dash, just write this. Blood, hair, saliva, semen, body tissue cells. Right? These are the samples taken there. Sometimes in the case of rape cases, then even the vaginal extract is also taken there to ascertain that really the person matches or not in that case. You know one celebrity case in case of Monica Lewinsky and Bill Clinton, that there's again proof was there. He was impeached also as a president, American president. He has to undergo impeachment. Again, all the DNA fingerprinting can be used ascertain those kind of individual identification part, right? So again, remember that what is very unique thing. It has to have cell. It has to have DNA. That part is there. Yes, even remember that. I'll be coming to that part. Just write blood, hair, saliva, semen, body tissue cells. Right. This all can be used for DNA profiling there. Right. And I'll come to that band pattern exactly. So what is you can see in the lab, the blood has been taken and then it is broken, DNA is taken out. They run on the gel, this electrophoretic device, and from there they try to ascertain what exactly the match is correct or not. Right. So I'll just come to this part. I wanted to come to that DNA band pattern there. I have already said in the class of Andy Tiwari case, right? In terms of identification case there was why both the samples are taken, because few of the DNA will match with father and few will match with mother. So in that case the match has to be from both the parents there. If the person is not the biological father, then the DNA will not match. Right? That's what is the ascertain in the case of DNA fingerprinting. Now, DNA fingerprinting is not only used in criminal investigation or paternity maternity suit or lawsuit or identification, but it is also used in case of dead bodies. Let's say a body which is recovered after a long time, right, from any, let's say flood or any kind of a fire, and then the person has to find out whether that person is the actual person or not. Sometimes bomb blast could be there. I'll give one good example, Rajiv Gandhi case. There in that case, DNA fingerprinting was done. So even it can be done for that purpose also. It's not only for individual identification. Now look at here this part. Let me ask there in this one. Now tell me that in this, was the suspect at the crime scene? This is a band pattern there. Suspect profile, blood sample from crime scene, victim's profile. Is it there or not? Very clearly is clear from the sample there. Just look at the blood sample from crime scene and suspect profile. Exact DNA match is there. The person is present, right? Now look at here this one. Now this one. Is this man the father of the child? Mother, child and man. So is that man the actual biological father of the child? Yes. Just compare one by one. You take child in the center, you compare. See, first two has matched with mother. Third one is matching with father. That man, right? Then fourth one also matches with the person. Fifth one is mother. Right? The last one matches with the person, that man. Right? Also matches with them there. So it proves that the man is the father. That part is there. This is what is done. After doing the law, all the run, this is how they'll take the photograph and then do the comparison one by one. And then comes to the part there, right? This part is it. So in case of paternity and maternity test, only few will be matched. So you can write here, if you say about application part, so first is what? Individual identification. Individual identification. Second, what I said is that paternity and maternity testing there. I'll give you more application there, paternity, maternity test. Do we have any materialistic society in India? There's a, generally comes in terms of father. Why? Because all the legal rights flow from father, inheritance. But there are society which is based on mother, materialistic society. Do we have such society in India? North state. Like I'll give you example of Meghalaya. In Meghalaya, Khasi, Jaintia, they follow this kind of thing. That's what happens there. Right, one of my friend, right, right, I'll tell you the story there. Vivek, not Kalia, Kaila. His surname is Vivek Kaila. Right. So I'll tell you what is there. when I visited his home, where JNU campus, so his mother is a professor in sociology department in JNU. So his father is a Muslim, right? He's a Muslim, and father is a professor in Jamia Islamia. So where I have to go? JNU campus. That is mother's quarter, right? And father was living there, right? They're two different religions, interreligion marriage. The mother is a Christian, and Vivek Kaila follows the Christianity, which is obtained from mother. And the father is from Kashmiri, he's from Kashmiri Muslim. He was living with the wife. They're in that wife's house in JNU campus. Right? This is what materialist society has got. That you have to follow everything from mothers. Surname, the ownership, land, everything flows from the mother side. So there what happened? जो विदाई होती है तो विदाई के समय में लड़कियां रोती हैं, लड़के रोते हैं पापा ससुराल जाना हो रहा है this is what happens, right? Once it happened, used to be very popular in Nayars in Kerala, right? अच्छी बात है ना, घर जमाई बनकर रहना कौन सा ऐड था हाँ हाँ हो सकता है Nayars they used to have this kind of thing. कितना दुख भरी बात होगा बताओ कि the boy is crying there, पापा आपको छोड़ के जाना पड़ रहा है and going to the bride's house there. This is what has happened, but very few you will find. Again, it can be a certain here, right? Now what I am writing here, genography. There is something called genography. Now I'll tell you what is genography. Gene plus geography. Right, you already have geography class. So where it comes? Anthropology. Again, DNA fingerprinting can be used in the case of anthropology research. Like for example, how modern man is related to Neanderthal. Again, same test, DNA matching, similarity and dissimilarity. That can also be done there. So what is genography? Study of flow of human gene across continents, geography. So how Indians are similar to Australians or Africans, right, Europeans, that kind of comparison. That is also done there. So genography is gene plus geography, flow of gene, human gene, right, across geographical areas or boundaries. Across geographical areas, this is comparison is done there. So there can be different ways of carrying out this test. So now we have lab in the, and now you understood that with this question which I have asked, which is correct or wrong. ठीक है यहाँ तक clear है? Now tell me here, where in India we carry out such kind of profiling test? Because the law will regulate the such kind of labs, procedure used in lab. So which is the standard lab in India where DNA fingerprinting is carried out? Dr. Alex Jeffries in the UK, he worked and developed that. But in India, we all samples where it goes, which lab it goes. In Hyderabad, there is a center, CDFD, Center for DNA Fingerprinting and Diagnosis. Let me write here. Center for DNA Fingerprinting and Diagnosis. This is the dedicated lab for this purpose only and supported by Department of Biotechnology in Hyderabad. You'll find in India yearbook also given this name, right? Center for DNA Fingerprinting and Diagnosis. So any kind of molecular diagnosis, diseases. So you can write here, what I've given, individual identification, patented matter says, genography, flow of gene. So fourth you can write also, inherited diseases also. This also we can find out there. Right, again we can find which defective gene has come from parents or any kind of profiling can be done. Inherited diseases. Again that can also be done there. Too hot, but that's what I'm also thinking that. आज लाइट चली गई क्या? कल से हो रहा, कल पता है मैं कल 3 घंटे ही सोया हूँ. क्यों? कान्हा का जन्म हो रहा था. Krishna Janmashtami. What happens yesterday? I don't know. There's some problem with the power system. I thought like it's Janmashtami, then lot of bulbs and systems. So there's a lot of load. So yesterday there was a frequent power cut in the night till 1 o'clock in the night. It happened in Rajendra Nagar also. No, That happened. It happened here also. Oh, that also I also suffered. I thought this area you had control supply. You had the same thing here. Oh, I thought because religious, मुझे लगा धर्म के कारण वो कन्हैया जी का जन्मदिन हो रहा है तो इसलिए ऐसा हैप्पी बर्थडे कर रहे हैं वहाँ पर. और कल तो ऐसा हो गया 12 बजे के बाद गाना ही बजने लगा. हैप्पी बर्थडे टू कान्हा एंड ऑल दिस वन. Near to that. Oh, let's take a rest. This is a, you know that global warming there. Okay, okay, guys. Since there is a problem with the power there, so since we had already had class for 2 hours there, only the 1 hour class has been there. So one way out is that we can go in this way that every class I can extend for 15-20 minutes extra. Instead of going like more extension, like 1 hour I require there, but not a problem there. I can go for, let's say, 15 minutes or 20 minutes each of the class, which will not be too much of tiring also there. And with that, we can make up the syllabus there. So is it okay with you, all of you? So we'll do in that way there. So let us go, because already for 1 hour, there's no power there. So let us continue in the next class there. And I'll go extra. Let's say we still have 4 or 5 classes. So at least they can go 15 minutes every class. So we'll be able to make up their part. Right? 20 minutes each class, we'll be able to do there, right? So let's work on that way. So we'll break the class here. And we'll meet in the next class there. And I will put certain documents on the online. Today, missiles and all those part which I discussed last time. Now you have this one, this biotechnology part, technique method. So I prepared a soft copy. I'll upload on the website there. Right? All this will come into. हाँ मिसाइल्स आ जाएगा आज मैं दे दिया मैंने मिसाइल्स आ जाएगा वहाँ पर ठीक है अच्छा टॉपिक फॉर नेक्स्ट क्लास जस्ट वन सेकेंड टॉपिक फॉर नेक्स्ट क्लास विल बी बायोटेक्नोलॉजी एंड हेल्थ केयर पार्ट ऑफ दैट